Uh, Good morning, students. Today we will start new chapter, that is principle of inheritance and and variations. In this chapter we will start from the topic Mendel's law. In class 10th you are uh, already studied about the Mendel's law. He gave three laws: law of dominance, law of segregation, and law of independent assortment. Okay. In law of dominance, you know that it is a monohybrid cross. Monohybrid cross means one pair of contrasting character are crossed to each other. In monohybrid cross, law of dominance is proved by the Mendel. When Mendel take same character of opposite alleles means contrast. For example, color of the flower. The color of the flower, two colors, or maybe height of the plant means tall or short. So when he cross to each other, red flower uh, plant and white flower plant. So in first generation, he found only red flower. In first generation, he found only red flower. Then law of dominance in monohybrid cross. Red flower when crossed with white flower, he found only red flower. When he the redness and whiteness of the flower are allies to each other. So this is the F1 generation. White color he never seen. In Mendel's explained that due to the character which is found in F1 generation, it is one of the parental character. So that character is called as dominant character. Clear? Second is law of segregation. Law of segregation, same law of segregation. When Mendel selfing this F1 generation, he found red, red and white. Means three types of genetic constitution, which is also known as genotype. And two is observable character, that is red and white. Ratio is 3 is to 1 for phenotype. As you already taught by me in class 10th, the character which we can observe, they are called as phenotype, and the character which is decided where the genes, types of genes are present, this is known as genotype. The genotype in F2 generation ratio, phenotypic ratio is 3 is to 1, and genotypic ratio is 1 is to 2 is to 1. So that is the monohybrid cross. Law of segregation. The definition of law of segregation is when a pair of allelomorphs are brought together in the hybrid. If one, they remain together in hybrid without blending them. So it also proved that purity of gene. As you see that here, uh, 3 red and 1 white. Hmm? The capital R and small r in this combination, you can see that one gene is from red and one gene is from white, but they never be blend together. It forms a red, and red is a dominant character. Clear? Third law is law of independent assortment. Law of independent assortment. So this law is depend upon two pairs of contrasting character. When Mendel's cross together two pairs of contrasting character. Law of independent assortment, that is two pairs of contrasting character. It is called as dihybrid cross. For example, height of the plant, height and color of the flower. The height is two types, tall and short. Color red and color white. Cross each other. In F1 generation, there's a mixture, but all will have dominant character. Tallness is the dominant over shortness, and red is dominant over the white. So it is F1 generation. When selfing of this F1 generation, in F2 generation, some of the characters are two combination. Here is two combination of parent generation, that is tall red and tall short white. But in F2 generation of dihybrid cross, we will see that tall white and dwarf red, two more new characters, new combination. So this shows that uh, in this cross, these pairs uh, show itself they are independent. They segregate independently of each other and produce uh, and combined with others. So it forms 16 it is very time taking to form this uh, cross. I will show you the figure from the book. See, that is four. Here is another two types, not tall, not red. It is the round and yellow seed. But you can see that in dihybrid cross 16 offspring will made, and these out of 16, 9 will be from dominant character generation, phenotype generation. 9 is to 3 is to 3 is to 1. 9 of red and tall. 3 is tall and white. Next is dwarf and red. Only one is of recessive character, that is short and white. So that is the phenotype. And genotype is 1 is to 2 because genes are separate. 1 is to 2 is to 1 is to 2 is to 4 is to 2 is to 1 is to 1 is to 1. So that is the genotype ratio. It is given in your book because see this one, the genotype ratio. It is in your book. When two pairs of independent alleles are brought together in the hybrid one F1, they show independent dominant effects independently. So limitation, the law of independent assortment applies only to gene pairs or different pairs of homologous chromosomes. Now some basic things, terms which you have to know about, which are used generally. First is crossing techniques. There are two types of cross. Punnett square. Punnett square, you know that we have to make this type of square. This is red and white. We have to cross. So gene of uh, sorry, gametes of red is here, gametes of white is here. Okay. Now cross, make the combination like this. Clear? So this is F1 generation. Homozygous when both genes are same. Heterozygous when genes are different. Monohybrid cross when one pair of contrasting character are crossed to each other. Dihybrid cross when two pairs of contrasting character to each other. Now test cross and back cross. Back cross when F1 generation is cross with parent. So that is back cross. Okay. And when F1 generation is cross with recessive parents. So that is test cross. So these are the terms which are generally used in alleles. Alleles are um, alternate form of genes. Okay. Genotype by रीजन ऑफ मेंडल सक्सेस. मेंडल के सक्सेस का रीज़न क्या था? बिकॉज मेंडल्स won the Nobel Prize after his death. The success is he chosen a correct plant by luck, uh, and he kept complete record of every cross, and he took one or two traits at one time of his experiment, and his experiments has large sampling size which gave greater credibility, and he was also fortunate enough that the characters he chose for his experiment did not show linkage. That is the most uh, important reason for Mendel's success. Okay, by luck, he has chosen those characters which has no linkage together. And when he started his experiment, so he uh, gives the pure line characters. After Mendel, so many experiments have done by scientists, and some exceptions are also came in existence. The exception of Mendel's principle is first incomplete dominance. That is incomplete dominance. Mendel's laws is dominant. According to law of dominant, when only one character will show in F1 generation, so that is dominant character. But in exception of law of dominance, there is incomplete dominant. Incomplete dominant, when one pair of contrasting character are crossed to each other. So in F1 generation, the offspring will have a mixture of these two characters, blending. For example, in snapdragon, it is a plant, dragon or dog flower, which scientific name is anti antirrhinum. ठीक है? So they bear pink flowers in F2 generation, F1 generation. In F2 generation, they segregate. Okay. For example, red and white. Okay. Red and white. When cross each other, according to Mendel's law, it should be red. But here it is pink. Means ki red is mixed with white and makes it pink. And in when it's selfing of this, that is F1 generation, that is parent generation. So it gives red and gives red, pink and white. It shows segregation also. And here is uh, in place of phenotype, here is different in phenotypic generation of Mendel's cross. In phenotype, it shows 1 is to 2 is to 1, and in genotype is also it shows 1 is to 2 is to 1. According to Mendel's law, it should be 3 is to 1. Clear? So that is the incomplete dominance. Next is codominance. Codominance means when both the characters shows in their offspring. So for example, डोमिनेंस लॉ ऑफ डोमिनेंस के ही ये एक्सेप्शंस हैं. कोडोमिनेंस, that is ABO blood types. ABO blood types in human being, it is a also known as autosomal dominance. They are controlled by the gene I. For example, gene I has three combination IA, IB, and this one. Clear? It has three alleles. We can say that. So IA and IB produce a slightly different form of sugars, यानी anti जो भी कहिए, and this will not form any sugar. So when IA and IB express both together, cross together, so it express IA and IB both. Any two of the three alleles are dominating, expressing, they are expressed together by blood group. If father has blood group A and mother has blood group B, so the offspring has blood group A and B both. As you know that due to these three alleles, blood group is four types: blood group A, blood group B, blood group AB, and blood group O. तो कोडोमिनेंस मीन्स कि जब साथ साथ दो characters show हो रहे हो, तो that is called as codominance. Here is A and B, both are dominant over this gene O. For example, if father has AO and BO, means one allele has A, one and other alleles has no, uh, and one allele is B and second one is this type. So it gives four types of blood groups. If they have four children, so they can be four types of blood. So, but this combination, when A and B only, so they forms, it means कि they have A and B both antigens. Next is paleotropy. Paleotropy means when when gene to influence more than one trait is called as paleotropy. For example, star synthesis in pea seeds. Star synthesis in pea seed is controlled by one gene. If it has two alleles, capital B and a small b. So as you know that same gene is called as homozygous condition. Okay. And another homozygous condition is this. So after the maturation of, if starch synthesis and produce smaller starch grains, so after maturation of BP seeds, they they are round and it becomes wrinkle. So it means that the heterozygous produce round seed and B is dominant allele, but the starch grains are intermediate size. So starch grain size is considered as the phenotype. So the alleles show incomplete dominance. That is also known as incomplete dominance. In this case, also an example of paleotropy, since same gene B is determined more than one trait, that is seed shape and seed size. Clear? Heterozygote that it produces round seeds, but the starch grains are intermediate size. If starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. So here is multiple alleles, same as again we have to turn over the ABO blood type. ABO blood type, multiple alleles. A well-known example of multiple alleles ABO blood type in humans, as you know that Landsteiner uh, was the discoverer of ABO blood type, A type and B type. The three different alleles are in blood group, as you know, A, B, and we can say that O. A allele form antigen A and B allele form antigen B, and I will not form. In this, both A and B are dominant over I, so that is known as codominant, as you know that. So we can say that ki only genotypes of various blood groups indicate that they are inherited in the simple Mendelian fashion. But parents' blood groups of their children can be predicted as different. Any two alleles can determine the more than one characters. For example, जैसे अभी आपको बताया था कि फादर इज ब्लड ग्रुप ए एंड मदर इज ब्लड ग्रुप ए बिकॉज ए इज डोमिनेंट ओवर ओ. ठीक है, वी कैन नॉट राइट दिस वन, we can write this one, and mother is like this combination, so you can cross this, A and this, B and this, form AB blood group, A blood group, B blood group, and O blood group. Any four types of. The blood groups of parents can possibility of more than blood groups. Next is polygenetic traits. Polygenetic traits means when it is qualitative characters, not uh, it is quantitative character, not qualitative. Because skin color of the human is controlled by several pairs of separate genes. The cross between black and white skin, so it will be depend upon how many genes are there. For it is also an incomplete dominance, example of incomplete dominance. For example, three genes, three shades for dark and three shades for light color. Okay. So when a person has three dark and three light genes, so that skin color will be intermediate color. For example, अगर हम ये कह रहे हैं कि three shade is for dark and this three is for light. Okay. So if any person has this combination, it will be of intermediate color. But when any person has more dark colors, so it this skin will be more darker than other. If any person has more this genes, their that skin, that person has more light colored skin. So next is pedigree analysis. Pedigree analysis means कि when we show the crossing by the symbols, not by the that. So in this pedigree analysis, male is shown by a square and female is shown by circle, and this is known as couple. Clear? And when we dark this symbols, it means it is affected by the gene. ठीक है, full and half. तो It occurs in every generation unaffected है, so it has no shade. So now it is a half part of this chapter, principle of inheritance and variations.